And you are ready to go.
And then if somebody asks a question from Auburn or something, I will hear them?
Yeah, you'll hear it, and you should see a bubble will come up at the bottom.
Okay.
If you don't see it, a lot of the time somebody else will say, hey, there's a bubble.
But it's going to be your job. Bubble alert.
But right now, unless you have any issues, I leave everybody unmuted. Mm-hmm. It just depends. Lately, I have not had any issues, so I've been leaving everybody unmuted, and they should probably, if they have something to ask, they should be able to just say it.
Okay, yeah.
But you can, if you wanted, in case you're wondering, if you wanted to, under attendees, see this?
Yep.
When you start, you can say, I'm going to mute everybody, and then when you want to do Q&A, you can just unmute everybody and say, okay, does anybody have anything to say or whatever, and then unmute everybody again.
Okay.
Completely up to you.
Oh, yeah.
For right now, they're all unmuted.
Yep. If somebody is like crinkling with the chips
correct.
right next.
You got it.
Yeah.
You got it. That's what ends up happening a lot, especially at the lunch ones, is the people coming in, they're eating, and you can hear them eating. Yep.
All right. Well, thanks.
Absolutely.
Should be good to go then. Speaking of chips, should eat my food first. Hello, ladies. There's lunch in the back if you want to grab any.
Great.
You do look very summery.
Yesterday was just what? It was supposed to be 70 and supposed to be, like, I think it reached like 77.
Mm-hmm.
So I was like, well, now I learned my lesson of wearing something warm when it's warm out.
Layers is where it's at.
Yeah.
Excuse me, where's Penny Jesser's presentation today?
I don't know.
You know?
I don't know that Penny had one. What is the topic?
On-for clinical trials.
I've
got, it was today, but I've got the wrong location, obviously.
Yeah, I think so, because I'm here, and he's not, and I know Penny, and I would have seen her.
I'm going to take mine.
Yeah. Somebody trying. Hello. There's some lunch in the back if you want to grab some. Good thing we're kind of informal here.
I'm really good about that.
So I need to meet with President Watts later. This would not be good to do in front.
Probably not. I'm glad you're getting it out of your system now.
Exactly. Yes.
Very fresh.
Yeah.
I do remember sometimes the chest cancer looks quite snazzy.
Oh, yeah, there are two.
Hello.
Oh, thank you, Dee. Hey. Of course. It was thinking about it, like, no. I was just here.
take it off. It depends on the flare of the skirt.
Yeah.
Whether there is zipping involved. Monday was going to be raining, so.
Do you ride at G7 Research and park it at Chick-fil-A?
I try, when I do, I park it at the corner of University and 20th. In Oaxaca.
Yeah. That one's not hard.
No.
Yeah, that's actually a better one because I was telling Nina I
Mhm.
want our team one day, like as an activity,
Yeah.
to go and get on the zip and like ride to the zip
Yeah.
somewhere,
Mhm.
and ride back. We don't think that Watson and Roxy would do it, but Nina and I would be.
Yeah. It's fun to go downhill.
I know. Yeah, but it's riding back uphill, but that's perfect because we can park at the bottom of the hill and walk up.
Yeah.
Do you know Nina?
Not yet, I think. Hi, Micah. Mhm.
Nina.
Sorry, yeah, I have like, grease all over my fingers.
She works in external affairs with
Oh.
Roxy and Watson.
Okay. Oh, wonderful. Nice to meet you. I'm glad you're here. Mhm. Hopefully I can educate y'all about all the crazy things that are going on. You could just shout a term now and then and sound intelligent is what I do. I thought it was green. SAR.
I'm starting to get most of the acronyms down, but
Mm-hmm.
there's still a few. I'm like, what? Okay, cool.
For those of you that are remoting in, we have a few more people grabbing lunch, so we'll get started in a few minutes. Let's see. There we go. I am. Mm-hmm.
Where in Holland?
Groningen. I went to school there, and then my parents are from Meppel, which is a teeny tiny little town, just an hour south there. Mm-hmm. Are you familiar?
Well, yeah, my family is from Holland.
Oh yeah.
Weersma.
Okay. Mm-hmm.
But unfortunately, they moved to the United States, and now my mother's German, so I speak German,
Okay.
and no Dutch, unfortunately. But on KLM, everyone knows me because of the Wiesma
Yeah.
last name.
Yes.
So I know Hoyer Cracker. That's it. So
Yeah,
I can
yeah.
get past the KLM.
Yeah, yeah. It is always kind of nice whenever you go to the game.
Yeah.
Yeah. This newspaper.
Yeah. It's hard for me though as a German because
Yeah.
but everyone's like so excited and I have to break it to them that I'm German.
Yes.
I'm American Dutch.
Yeah. I've been here almost 15 years.
And then going to.
Sometimes people
Then
like,
going back to lab.
working with because I'm pretty sure I do. And then a Canadian, a friend of mine, his mother come visit me, he's kind of laughing like, if I can make sure with that culture that Texas, that's horrible. Well, thank you, you should talk.
Well,
Yeah,
it'd be fun to get a picture of
Yeah. So you already
you say
completed
all,
the
it's a
grant.
good word.
They're helpful.
Yeah.
It does, it's very multi-purpose.
He already prepared the presentation, we practiced one with his lab members, and we practiced yesterday. So it should be fun.
Is anybody else here now still? No. Okay. All right. I think we're going to go ahead and get started. Thanks everybody for remoting as well, and hi everybody that's here. You're enjoying a nice lunch. I hope everybody on the computer also has their own nice lunch. You're currently on mute. If you eat a chips bag right next to your microphone, I may mute you, but other than that, we should be in good shape. And whenever you have a question, just go ahead and ask it. So today's the first lecture in a series that we're going to have over the next couple of months, where I designed it in a way because I found out after we started the Alabama Drug Discovery Alliance and what that alliance entails, we'll talk about a little bit today. Talking to people on campus, there really is a limited knowledge about the drug discovery and development pathway. In some of the graduate courses, we do have an introduction about that, and we talk in some more detail in a few classes. but for the majority of people, especially when you're already faculty or you're postdoc or whatnot, you don't have the graduate school classes available to you. It's like, well, how do you actually do this? You write a grants and you write papers, and then your last sentence in your abstract is like, am I going to cure cancer? Or something like that. Well, how do you actually do that? So I thought to put a lecture series together where we highlight all of those steps along the pathway with a little bit more in-depth educational talks instead of, sometimes when you have drug discovery talks, people kind of talk through the whole process and they're amazing and they've cured HIV or something like that in an hour. Well, I want to kind of drill down into how does a chemistry program work? How do you actually apply for an IND? What's pharmacokinetics entail? Things like that. So today is really going to be an overview type of lecture. I'll breeze through some of the concepts maybe a little bit quickly because we'll have an in-depth talk about that particular concept, but I want to kind of set the stage about what the whole pathway looks like and also talk a little bit about how do you get money to do this kind of research, because it's not typical R1 driven research that you would go to NIH for. How do you actually find resources to help you with that? So we have some local resources that I'll talk about as well. But I'll start with kind of the concepts and the overview of how drug discovery works. So trying to get to the next page, and oh, there we go. Here we go. So this is a graphic that I got from Nature Drug Discovery about the pipeline. It's kind of an overview of how this process works. And it's a pretty long pipeline. When you look at the timeline of going from the ultimate beginning, which is target identification and what that means we'll talk about, to ultimately approval by the FDA, typically you're looking at 12 to 15 years. So this is not an instant gratification program. This is let's focus on the long-term goal. But these are kind of the steps that in the early phase here on the left side of the graph, we have things like target identification, hit generation when you use high throughput screening, and lead generation. Again, I will talk through these concepts so you know the terminology, so that if somebody comes in and starts talking about, these are hits and these are leads, that you know what we're talking about. Then just really in a broad schema, after this first initial phase, and this is the discovery phase, we typically do animal models to test whether the molecules that we found actually treat whatever disease you're looking for in mice or rats. And then we get to a clinical development phase where we actually need to test it in human patients. And we have several phases in that, and we'll go through those. And ultimately, you need to go to the FDA and submit your packet. Look, this is the risk to benefit ratio. Can we sell this? Yes or no? So today I hope to kind of touch on each of these particular points. So this is kind of the schema that we'll walk through today, all the way from target identification at the beginning to regulatory approval at the end. So let's start with the beginning, which is almost the most important part. It's harder than you think. So we have a whole lecture about what is a target and how do you identify that and how do you validate a target. So what is a target? The definition, let's start with that, is the naturally existing cellular or molecular structure involved in the disease that a drug in development is meant to act on. So it's really on the molecular level, what does your compound bind to and manipulate, either inhibit it, activate it, block it, whatever it needs to do. So you're really looking for maybe a DNA sequence, you're looking at the amino acid sequence and the protein, things like that. So in order to define this, you really need to understand the molecular mechanism of the disease. And then within that mechanism, you need to identify that particular target. So many times people, for example, have to find, okay, this enzyme is absolutely critical in, let's say, diabetes, and I want to increase its level of expression. So let's find a compound that does that. Well, you can screen for that by, using a reporter gene and just, talk about some ways in which we can screen. But then it's almost what we call a phenotypic screen. When you add a compound to a well and you see an increase in the expression of that particular gene, well, it does what you want it to do, but how does it work? What's that black box? What's happening in that cell that causes that compound to bind to something and then have that result of your gene expression goes up? So it's very detailed, and it's more detailed than people sometimes tend to think. But all this information really comes out of basic research, and that's where universities, of course, shine. And that's why doing drug discovery in an academic setting has, a lot of attractiveness, because we have the experts on many disease processes on whatever disease you're interested in, whether it's Alzheimer's, tuberculosis, kidney disease, you name it. Let me actually minimize this real quick. There we go. So you have identified, okay, I want to manipulate this particular target, this molecular structure. Well, how do you know that's actually a good idea? So that is what we call target validation. So this is actually quite difficult because ultimately you don't know whether something actually is going to work until there is an approved drug on the market that treats patients. It's kind of tongue-in-cheek almost, but it is the reality because many times we have cured cancer in mice so many times. I mean, it's almost pretty easy, right? So it's a translation to the human situation that really makes or breaks, is this target validated, yes or no? So you start, as simple as possible to answer this question by first doing some experiments in cell culture. Let's say you have identified, okay, this molecule, if I find a compound that inhibits the activity of this molecule, we're going to, cure asthma or whatnot. Well, maybe you can use some molecular biology techniques like shRNA or something like that to look in a cell culture system, does it do what we want it to do? And if that answer is yes, then you go to typically mouse or maybe rat models and see in at least a human, a whole organism, something that lives and breathes, does it still work? If the answer is yes, well, that adds value to your target. If there's any clinical data out there, maybe people, there's a lot of databases, for example, in cancer where there's tumor tissues and they compare it to healthy tissues and they look for expression profiles of genes or proteins and they look for differences. Well, if your target is different in normal versus the healthy situation, normal versus a disease situation, well, that gives you some indication that it does play a role in your disease. But then ultimately the best validation is that there is a drug that works in humans. And that's what we sometimes call me too drugs. So if one company has come out with, a particular molecule that hits a certain target, then you'll see many copycats that they will find their own chemical matter, their own compounds, but they do the same thing because now there's proof that when you manipulate the target, you're going to have an effect on patients. So you have de-risked the target then by this point a lot. So how do you then pick which ones to go after? So there's a bunch of criteria that we look at when we decide to invest. And there's a whole seminar that I'll talk about in more detail about next week. Usually Mark Sudo, the Vice President for Drug Discovery at Southern Research, gives this seminar, but he was not available. So he let me pretty much use the slides. I had to promise to buy him a burger at, Café DuPont or something like that. But other than that, I can use his stuff. But this is just really briefly some of the things that we'll look at. Is it novel? Especially in an academic drug discovery setting, we don't want to do something that, let's say, Pfizer is already doing because they have much more money, they have a lot more personnel that are devoted to these things. We really try to come up with what's the new and great information that we as an academic institution have access to that is maybe a little bit too risky for a more established pharmaceutical company to go after. Is it what we call druggable? And this is a term that's hard to define sometimes, but it's typically used as it has this class of molecules been targeted by drugs before. So for example, kinases. Kinases play a role in many disease processes. We know that there's an active pocket in a kinase and there's a few hundred kinases, and we have compounds that inhibit selective kinases, only certain ones and not others. So we know there's kind of a paradigm out there that we can do this. Something that's a little harder to do, a lot of people have expressed concerns about this, is what we call protein-protein interactions. Can you disrupt kind of the binding of two proteins together? And proteins tend to bind to each other with a lot of contact points, so you can kind of imagine that the small molecule has to kind of wiggle its way in between there and then push them apart. Well, is that something that a small molecule actually can do? We have some evidence that it can, but it's a little bit more risky compared to what's out there. We also look, here's the last bullet point, on intellectual property potential. Ultimately, we want to find a molecule that has not been described before, because that way we can file for a patent for it. Now, you could say, well, we're an academic institution. Why do we care about making money? I mean, that's for greedy people and, pharmaceutical companies. Well, we'll talk about how much it costs to develop a drug. We're talking about hundreds of millions of dollars. Ultimately, we will need to partner with the big pharmaceutical company because we need the money and the resources to do, let's say, large-scale clinical trials. If you want to ultimately develop a therapy that gets to a patient, you need to have a patent around something, because ultimately people that invest in this need to have a way to make their money back. So it's a very practical reason that we look for what's the IP potential around this particular space, because that's how we ultimately make therapies become a reality instead of an academic exercise. So we're not selling out or anything like that. It's just a reality of how drug discovery in the world works. So like I said, we'll have a seminar next week about this in much more detail. So we've identified what target we want to go after. Now, how do you then actually find a molecule that does what we want it to do? So there's a few ways you can go about it, and today I'm going to talk about high throughput screening. So the acronym for that is HTS, and you pretty much use a robot to screen through thousands and thousands of molecules to see if they do what you want it to do. Now, in order for a robot to be able to do that, you need to give it really clear instructions. And you need to trust that the robot will get meaningful data out of the protocol that you pretty much give it to. So in order to do that, you need to what we call assay development. So assay is a test that the robot is doing, and you need to develop the protocol in such a way that you will get data you can trust from a robot that cannot think and look at, you know, your wells with cells and be like, let's incubate it for a little bit longer. I mean, it doesn't have any judgment calls like that, right? So you need to have the same protocol that's being executed all the time in the same way. It's that assay development before you can do the HTS part. So the high throughput screening platform is really a system that uses a lot of different parts together. So it uses specialized automation equipment or robots. And then high density microtiter plates. And I have a picture of that in one of the next slides. So you're all probably familiar with 96 well plates in the lab that are about yay big. So the microtiter plates that the robots use are exactly the same surface area, but instead of 96 wells, it has 384 wells in there, or 1,536 wells. So you can imagine why you want a robot to do this, right? Because otherwise you're like, okay, 12 to the right and then 26 down. I mean, as a human, the pipetting gets kind of tedious. So robots can do that really precisely. And they can then screen a large number of these wells and these plates in a relatively short time. And throughput, when you have it set up of, you know, several thousand compounds per day, is very common. So there are a couple of parts to this whole system. One of them is compound management. So we work with Southern Research a lot, and they have about 1 million compounds in their freezers. About 750,000 are actually usable. Typically in our programs we screen 100 to 200,000. You want to be able to track which compound was in which well. From what plate did that come? What vendor did we buy it from and when? Well, how many times did that plate get taken out of the freezer, thawed, put it back in the freezer and frozen again? Things like that. So you want to have a tracking system that uses barcodes usually to keep track of that. And also when you have the data ultimately spit out in a huge Excel spreadsheet, it needs to coordinate the data that you get out of there with the compound that caused that effect. So you also need precision robotics for liquid and plate handling. Like I said, you want a robot to probably do your pipetting for you and not yourself, like 1 nanoliter in, you know, this particular well. So there's a few instruments in Southern Research that are really exquisitely good at manipulating liquids. For example, there's an instrument called the Echo. It is an acoustic liquid dispensing instrument. So what it does is you have a plate, you know, in regular side up, sitting on a machine that shoots an acoustic wave through the well from the bottom to the top, and that acoustic wave causes a little drop to fly out of the solution up to a plate that's hovering over it upside down. So, and it can dispense nanoliters precisely. So much better than we as humans could ever pipette. Of course, you need the informatics. Again, like I said, to evaluate the data, you need to associate the data with particular compounds. And you also want to do some cheminformatics. Cheminformatics means that based on the structure of the molecules, some kind of fall into a family of molecules. They all kind of look a little bit alike, but have slight differences. Well, we call those clusters. If you have a cluster that all tend to do the thing you're looking for, let's say it's inhibition of a particular enzyme, well, it gives you some information, and you can use software to kind of help sort through that data and not have to manually go through a spreadsheet of hundreds and thousands of compounds and with your eyes trying to figure out, you know, what makes sense. So there's software and cheminformatics that can help with that. And then, of course, you need people that know how to do this. And Bob Bostwick is the director of the high throughput screening lab at Southern Research, and he will give a talk on March 21 on how exactly this happens at Southern Research. And there's really wonderful infrastructure there. He came from AstraZeneca, and has done many high throughput screening campaigns in his life, so he's really the expert on that. And this is a picture of the microtiter plates I talked about earlier. So here on the left is a 96 well plate. You can see the volume that you're probably used to manipulating, 100 to 200 microliters just with a regular pipette. You're going to be accurate enough. But then you have your 384 well plates, 1536 well, or you even have 3456 well plates. We're not using these at Southern Research, but even the 1536 well plates we do quite regularly. And what the advantage is that your volume is much lower. And when you screen a lot of compounds, many times your reagents to do your assay are quite expensive. So the smaller volume you can do things in, the more affordable it gets. So this is kind of generally how we approach a campaign. So an assay gets developed, typically in the PI's lab. The PI can be either UAB or Southern Research, and they kind of have it working in a 96 well plate format. It gets then handed off to Southern Research, and that lab optimizes the conditions and the protocols that the robot can execute it. And once it's kind of, in good shape, then a test set of only 10,000 compounds gets run at 2 separate times on 2 different days. So you test in duplicate, and then you kind of look for the correlation to get the same data on both days. If that looks good, then we initiate the full screen. So typically we screen between 100 and 300,000 compounds. And we only do one compound one time, N of 1, and that's why you need to be really sure about how your robot does it.
So you said you had 750,000 compounds.
Mm-hmm.
Your typical screen is 100, 300,000. How do you pick which ones to try?
Yeah, so how do we pick the subset to kind of throw in the screen? So we do that in consultation with the chemists, and there's a couple of and I'm sure Bob will talk about what's all in their collections, but sometimes you can buy a collection that's dedicated to a specific therapeutic area. For example, these molecules are more likely to penetrate the blood-brain barrier. So if we have a CNS project, that's the one we would screen. Or these are molecules that kind of look like they will be kinase inhibitors. So if you have a kinase as a target, we're going to throw that molecule in. We also have a collection of FDA approved and unapproved drugs, and we tend to screen those now actually all the time. So the FDA approved drugs is kind of obvious. There's 1,500 or so drugs out there. But then we also have about 1,500 what we call unapproved drugs. So they were in clinical trials, and they maybe failed for a particular indication. They were not useful in treating arthritis or what people were looking for, but it was not because of toxicity, but just because they didn't do what they were designed to do. But sometimes biology kind of catches up, and then it realizes, hey, wait a minute, the same mechanism may actually be of use in blood pressure regulation. Let's actually kind of see if the compounds that were already developed, and it seemed to work in humans, it just didn't treat asthma. Let's look if they actually treat blood pressure, things like that. So we have that collection as well. So yeah, together with the chemists, we decide which ones to screen. So when we do the screen, typically we identify 2,000 hits, or sometimes we just call them actives. They do something. But then which ones, how do you rank those? So what we then do is these were all at this point screened at one concentration one time. What we do now is we cherry pick, this is kind of that's a technical term actually, you would not think so, but we kind of, from all the different plates, we grab the ones that were active, we put them on new plates, and then we screen them in dose response at 10 different concentrations. So you create IC50 curves. So typically if something were active, the higher the concentration in the well, the more of an effect size you should see. So typically you get a sigmoidal curve, and then you can calculate at what concentration do you see 50% of my effect. It's called the EC50 or IC50. It kind of gives you like which ones are more strong, more potent than others, and you can rank them. And then at that point, we many times also do sometimes a counter screen. So let's say you are a kinase screen is a good example. You want to inhibit kinase X, but not kinase Y. At that point, you can kind of screen them for both at the same time in concentration response, and you get 2 numbers, one for how good it is to inhibit your kinase you want to inhibit, and the one that you don't want to inhibit. So you can kind of rank them that way and get some valuable information to then for the chemists to look at, and they will pick like, oh, this one looks interesting based on the profile of how they behave, but also then on the structure, what's the chemical composition of this. So the chemists will ultimately then generate a short list. And it depends a little bit on how many they pick, what happens next. Sometimes we still have quite a large number. We had one project, for example, we had 4,000. It's not like, oh, a Western blotting idea doesn't seem like so smart right now. So I mean, at that point we did another almost like medium throughput screen to kind of whittle down at 4,000 to even less. Many times there's still, the chemist can kind of rank and maybe pick between 10 and 50, and we order a new compound, and then the PI can test those compounds in their own lab. So then you have even more confirmation. It works in Southern Researchers' high throughput screening lab. It also works in the biology lab here at UAB. Now we really have a lot of confidence that this does what we want it to do. So we always do want to reorder at that point, fresh powder. We also now always confirm that the structure is what the manufacturer says it is, because we've run into a few issues where it was not the case, and then you can go down a rabbit hole and we just had the wrong information what the compound looked like. So we retest them, and then you kind of start an iterative cycle. So the data goes back to the chemist. They start to hone in on, okay, these 2 or 3 what we call scaffolds or types of molecules look most interesting, and then they start maybe synthesizing some analogs around them, and analogs are kind of brother and sister molecules of the compound that you're looking at that maybe change an atom at a time from a carbon to a nitrogen, or instead of a 5-membered ring, they put a 6-membered benzene ring in the molecule, things like that. I'll talk a little bit more about chemistry in a few slides, but before we leave high throughput screening assays, I did want to point out to the 2 classes of assays you can do with the robot. So there's one that we call cell-based assay, and it's kind of what the name implies. You grow cells on the bottom of the plates. So many times this is when we use 384 well plates, because there's enough space for quite a number of cells to kind of grow on the bottom. And the advantage of using that kind of an assay, where maybe you add compounds to the cell and you look for a fluorescent signal, or maybe cell death or things like that, you know that if you see an effect, that the compound was able to penetrate through the membrane typically, or get into the cell and do its effect. And sometimes cells need to metabolize compounds in order for the effect to become apparent. Nucleosides and nucleotide analogs are a good example. They're taken up by the cell and they're phosphorylated inside the cell, and then they get intercalated in the DNA, things like that. If you would just add the compound that ultimately does the trick by messing with the DNA, it wouldn't even get in the cell. It would be, the membrane would be a barrier. So cell-based assays have that advantage that you kind of raise the bar a little bit. The compound needs to do a few additional things in order to do its effect. The negative would be that sometimes you then will throw out compounds that would actually work with a little bit of tweaking. Like getting the compound to be taken up by a cell is something that a chemist can probably fairly easily do. So you may, shoot yourself in the foot a little bit by raising that bar a little bit. Now the other main concern is that it tends to be more expensive. I mean, just for fun, look up the cost of a bottle of FBS for change, and you will fall out of your chair. And now think of having to treat hundreds and hundreds of plates with medium that uses FBS. You can imagine that the costs start to add up pretty quickly. And it also is a more complex system, so it's a little bit more variable. The variability, high throughput screeners don't tend to like. They like everything to be precise and the same. So the alternative is to do a biochemical assay, and that's typically just an isolated system in a buffer solution. For example, if your target is an enzyme, you can purify the enzyme, just have the enzyme there, and then have a substrate, and then your compound, and you look for the effect, whether that's fluorescence or fluorescence polarization, or there's a couple of ways that you can design these assays. And Indira Patmalayam is the head of the assay development and lead biology group at Southern Research. She will talk March 7th about how to design this. So the advantage of doing a biochemical screen is that if you see an effect, you know your compound works on your target because, well, you see an effect, right? It's also cheaper. But the negative is, well, you know your compound messes with your system, but what else does it do? I mean, bleach kills a lot of enzyme activity. I mean, but you don't probably want to use bleach to treat your disease, right? So typically what you do is you use a combination of these two at some form or fashion in your what we call compound progression pathway. So it's kind of a roadmap of which types of screens you do in which order. So you may do a high throughput screening assay on 200,000 compounds in cells, and then you can maybe do a follow-up screen in the lab on a smaller number of compounds that show activity in the biochemical assay to know for sure that your compound that had the nice effect in the cells actually is messing with the target that you want. Or vice versa, you do a biochemical assay okay, we want to inhibit this enzyme function, but then you may do a cell-based assay to look for general toxicity, and you throw at anything that's like bleach and kills all the cells. So it's not like you always need to do one or the other first. It's just whatever makes the most biological sense. So this is done in a lot of brainstorming with the high throughput screening group, with the biology expert, and you kind of come up with the best plan. So ultimately, assay development is a big bottleneck though. So this is a chart I plucked from somewhere on the internet. You can find anything on the internet, including drug discovery stuff. It's amazing. So everything in blue is fairly easy, right? Compound libraries, you can buy them, right? Target identification, well, just go to PubMed and type in the latest diabetes paper, and you'll probably find somebody's claiming that this is a new target and we're going to cure diabetes with this. Well, the hard parts are in gray. So target validation, how do you know that's actually a good idea? Talked about that. But then also assay development, this is actually quite difficult to get something from an idea, something working in the lab, to having a robot execute it. Then again, once you get to, you've optimized and the robot can execute, it's pretty much push the button and you get reams and reams of data. That's also the easy part. But why is this assay development a bottleneck? Well, it's because when you go from manually pipetting to, you know, the robot executing things, you really have a couple of different rules to play with. So what you're aiming for in HTS is to have a good chance to believe the results of an N of 1. One well, one concentration, one compound. So you really need to have exquisite reproducibility between wells, between plates, between days, things like that. And that's why that is such a long process. You're like, oh, come on, here's the protocol. Why don't you, go run it? But honing in on making sure it's super reproducible, and no matter where your compound is, if it's on the edge of a plate or in the middle of a plate, on a top stack of a thing that's shut in an incubator, or if it's in the middle, it should give you the same result. That's why it's so hard. So look forward to the HTS and assay development seminars about that. So then once we've identified, okay, these compounds seem to work, well, what do we do then? Then we start involving a chemist. So if HTS is kind of like a funnel, you hone in on a couple of molecules that seem to work. The medicinal chemistry program, I don't have a good analogy for it, but you pretty much start to tweak molecules in as many different ways as you can and look for, is it still effective? Does it get more potent? Is it still selective? Does it get metabolized by the liver in 2 minutes or half an hour? Things like that. So there's a lot of tweaking there. And Corinne Ojeli-Zeffran, she's the director of chemistry at Southern Research. She will give a talk about chemistry on March 14th. I'm not a chemist, so I cannot do it justice. And she's a fabulous chemist and actually can explain things to non-chemists, which is not trivial. So what you want to do in the chemistry program is establish SAR. So that's another one of those acronyms that people throw around a lot. So that stands for structure activity relationship. So if you change the structure, what does it do to the activity of the compound? So that's where you start to purchase or synthesize analogs, so this kind of sister and brother molecules with slight tweaks, and you test all those analogs for efficacy, selectivity, things like that. And then the data is used to go back to the chemist, and they will design a new round of molecules, and you kind of go back and forth for quite a long time. And ultimately, if you have a profile that kind of fits your parameters, you're like, okay, this is looking pretty good, like what I'm needing for ultimately efficacy, that's when you will start to test those molecules for what we call ADME or ADME, which stands for absorption, distribution, metabolism, and excretion or elimination. First in vitro, and then ultimately you can also do that in animal models in vivo. And I think I have a slide about that. And you also want to look at the pharmacokinetic parameters of these molecules when you give it to a mouse or a rat. How long does it circulate in the bloodstream? Does it get excreted through the liver, through the urine? Do they breathe it out? Where does it go to? So typically you want to do this before you test for efficacy typically, because especially if you have long-term animal models where you need to treat mice for 2 weeks or a month, or sometimes you have 3 months. I mean, think of Parkinson's disease models or Alzheimer's or whatnot. Well, you don't want to find out after 3 months of treating animals that your molecule was never even absorbed in the body. I mean, that would be a waste of resources, right? So you always want to do your pharmacokinetic evaluation first. What does the body do to the drug? Does it hang around long enough to actually have a chance of doing what you want it to do? So there's a lot of parameters. Sometimes it's compared to a Rubik's Cube when you make those modifications to the molecules and you change one thing, and now you have increased potency, but oh crap, now it gets metabolized by the liver in 2 minutes. So everything changes everything, and so that makes it really challenging. So we're typically looking for potency. How does it get metabolized, physical properties, does it now get dissolved in water or DMSO, things like that. Is it now toxic? So we try to look at the whole picture as a whole, so it's quite complex. So then some of the ADME studies that we can do in vitro is, for example, will it get absorbed when you take an oral pill? It needs to be taken up through your gut into the bloodstream. And there's some model systems that can kind of predict to a certain extent whether it is likely or not. Of course, it's a model system, it's not real life, but you can kind of hone in on some things that way. So there's a permeability assay you can do with KCO2 cells. It's I think a colon cancer cell line that forms a really tight layer in a well, and then you can look at, okay, you add a drug at the top and you can see at the bottom how much went through, things like that. Metabolic stability we look at a lot. Microsomes are kind of the chewing up organelles of the liver. So you can use them from mice, from rats, from humans, from monkeys, from dogs, whatever you want to look at. You can kind of see, okay, let's incubate these microsomes, these things that want to chew up drug, with our drug into B, and let's see after an hour how much is left, or after 2 hours. And the longer it stays around, the better your chances that it will stay around in the body for long enough as well. And then there's a lot of enzymes it's called the cytochrome P450 family. They're pretty much the enzymes in your liver that detoxify whatever you eat and drink. And there's a whole host of them. So we typically want to see what family member are actually metabolizing this. And there's actually interesting drugs can also either activate the activity of those enzymes or inhibit it. So there's a lot of drug-drug interaction that happens this way, because if your drug activates one of the enzymes and that enzyme is also maybe metabolizing your daily cholesterol drug, then maybe your cholesterol drug is not going to work well anymore because it's being chewed up a lot faster, so it's not hanging around in the body as long. So again, pharmacokinetics is very complex and a lot of fun. Did you believe it when I said that? Actually, I got my PhD in pharmacokinetics. I kind of like it. Okay. So then you also start to look in vivo after you've cleared a few parameters in vitro. And you can, for example, look at where does it go in the body. You can radiolabel it, and if you're looking for something that needs to work in Parkinson's again or in Alzheimer's, does it go into the brain? Things like that. So ADDA Costa, who is really the pharmacokinetics expert here on campus, he will give a talk on April 11th, and he's given it a few times and is actually very understandable and very accessible, so I highly recommend his talk about pharmacokinetics and ADME. So then ultimately you start picking candidates. So again, we started with high throughput screen where we first had actives, and then if they're confirmed, they're called hits. And then these hit molecules get modified by chemistry and they become a lead molecule, and then lead molecules ultimately become candidates for clinical trials. But before you can administer anything to a human being, you need to do some additional testing. You need to do in vivo efficacy testing, but then also, and most importantly, and this is what the FDA will look at most, is your toxicity testing. So typically you need to do acute toxicity where you just give one dose to at least 2 different mammals, and look for what dose starts to give untoward effects. But you also need to do, depending on how you're going to use the drug, repeat dosing, especially when you're looking at something that treats diabetes, for example, or like Parkinson's disease, things that are long-term diseases. You're going to have to do long-term toxicity studies because you're ultimately going to give this to humans a long time in a row. And that needs to be done on both one rodent and one non-rodent. So typically dogs or monkeys. I kind of combine them. Donkeys are not used. Dogs or monkeys. But this is, you want to do your toxicity experiments as much as like what ultimately the clinical use of the compound is. There's not really a roadmap on how to do this. There's not a formula. This is something that you start to work with consultants. You start to talk to the FDA, and you kind of come up with the plan together. Because you don't want to make a plan and then go to the FDA and they're like, why did you do that? I mean, that will be a waste, right? So there's a new director of toxicology at Southern Research. I haven't met him yet, but he did agree to give a seminar. So I like him already. So Dr. Paul Bushdid, I don't know if I say his name correctly, is going to talk on April 25th about toxicology. So in summary, again, here's your Rubik's Cube. You change one thing and everything changes. You want to consider, do I hit the target? Does it work in animals? Is it safe? Is it not toxic? What's my pharmacokinetic profile look like? How do we need to formulate it? Do we make a tablet? Is it a solution? Things like that. And that's when you kind of talk with the FDA, this is the plan going forward, this is how we want to get it into humans. And it really depends on the indication, what tests you need to do and how you need to do it. But at this point, you need to do everything according to GLP, good laboratory practice. It's very expensive, just so you know. So the goal is then to get to an IND application. So IND stands for Investigational New Drug. And that's the approval or allowance of the FDA to first test it in humans for the first time. So the CCTS, which is co-sponsoring this lecture series, has a support office. So Penny, Jester, and myself are in that office, and we help people on campus all the time at IND applications. And we will present how to apply for an IND on April 4th. We're not experts, but we're kind of one-eyed in the land of the blind. So together we always figure things out, and we've helped a lot of investigators on campus on how to do this. Pardon? Mm-hmm. Yeah. Yeah, so in an IND application package, you have some background information on this molecule is treating asthma in my animal model. The FDA actually doesn't really care that much about whether it works in animals, because they're like, ultimately the proof in the pudding is it going to work in humans, right? So what they look for is safety. So there's going to be a GLP toxicology package in your IND application that documents exactly how much was given to which animals. Were they dogs, were they monkeys, were they mice, what was the dosing schedule, and what were their liver enzyme parameters, all that kind of stuff. So if it's a completely new drug that's never seen humans, that part will be several inches thick. Sometimes we help with IND applications where it's a repurposing application, where there's an existing drug, but we want to use it for something else now. So there's already a data package on file with the FDA. So then we go to the manufacturer of that drug and we're like, hey, we want to try your asthma drug in a blood pressure indication. Is that cool with you? And can you give us some free drug to try it? And they usually say yes. And then we say, can we get a letter from you that authorizes the FDA to look at your own data file? So it's called a letter of cross-reference or letter of authorization sometimes. So that means that they don't have to send us that data package. So that, you know, confidentiality is guaranteed. We don't have to see how they make it and all that kind of stuff. The FDA can just look at their drug file, drug master file it's called, and look at that data before they give us the okay, yes or no. So first in human, the INDs tend to be several inches thick. When it's a repurposing thing, it's, you know, half an inch thick. So very different. And they take less or more work depending on what it looks like as well. So then ultimately, I'll breeze through this because I think most people know this. You have 3 phases at first for clinical trials before approval. So phase 1 is typically done in healthy volunteers in a small number of patients. You slowly increase the dose and you look for, you know, what weird thing is going to happen to, you know, a healthy human volunteer. What you look for is absorption metabolism, so there's a lot of blood draws at certain time intervals. Do you have any gross effects on organs and tissues? What are the side effects as you increase the dose? Now, in some indications, let's say oncology, typically those drugs tend to have nasty side effects, right? So in that case, you actually do your phase 1 in sick patients. Same was when the initial HIV drugs came out. They also were bad molecules, but you know, they did what we needed them to do. That was all done in a patient population. But in your usual, you know, diabetes, Parkinson's disease, blood pressure type stuff, usually done in healthy volunteers, college students making a little bit extra money on the side, things like that. So then we go to phase 2. Now this is typically the first time a drug sees a patient. So you're really going to look for, does it work? And this is also where most drug clinical trials fail. It's fairly easy these days to predict, does it have the right pharmacokinetics, you know, does it not kill humans, you know, at a little bit, but does it actually work? Well, that is a really difficult thing to prove. So phase 2 trials tend to be expensive and they weed out kind of the good from the bad. You're also going to look for short-term side effects and kind of what is the dose that we need to be looking for to look at the desired effect. And this is typically then when you have a control group and a treatment group, maybe blind, double blind, placebo controls, things like that. If that still looks good, then you go to phase 3. Now typically you're looking at multiple clinical sites thousands of patients, and this is really expensive. And this is also where usually even at phase 2, you're going to want to partner with a company, for example, that knows how to do this, because it's not trivial to do these trials, and also has the money to be able to support this. So especially phase 3, this is typically nationwide. You need to have a coordinating center that keeps track of all of this. And here you're really looking for what's the benefit versus the risk package. And then what are some less common side effects that may pop up when you treat thousands of patients. And this then ultimately informs what's on the label. And the label is that insert in your package that's his teeny tiny print that says you're going to lose your hair and be nauseous and all that kind of stuff. That comes from the phase 3 clinical trials. So then you go to the FDA, or if you're in Europe, to the EMEA, or in Japan, there's a Japanese agency, et cetera, and you submit for regulatory approval. Unfortunately, this is not worldwide coordinated yet. There are efforts to kind of streamline this so that you can submit the same package at least to the different agencies, and you don't have to reformat everything on their particular forms and things like that. But it's still a process that typically can take a year or longer, just because it's so much information. And then you hopefully get approval. Well, then you're not done yet. It looks like it. But no, you still need to do what we sometimes call phase 4 clinical trial. So you're actually really never done. Phase 4 is really safety monitoring. So if there's toxicity, if weird toxicity that pops up, there's actually a form the clinicians can fax into the FDA, and it goes into a master file to kind of keep track of, until you're in the real population, the real world, sometimes you really don't know of any side effects until that really is the case. So the FDA or any agency can still pull your drug from the market even after it's given approval. And this is also an opportunity, though, to go after additional indications. So if you, for example, I've worked with somebody who's in the Prader-Willi syndrome foundation. That's a disease, it's a genetic mutation that causes patients to have insatiable appetite, so they pretty much eat everything in sight. There's a lot of developmental delays with that as well, so you have to literally lock the fridge and the cupboard and things like that. So there's a lot of companies that are going after that particular population for an initial approval, because that's an orphan disease. And there's a lot of incentives to find drugs for indications like that. But then if it gets approved there, then you may go to the obesity market, because your hurdle is going to be a little less because you already showed that it's safe in patients and things like that. So sometimes that's kind of a strategy in many different disease areas. Go for one kind of niche population first, because it's easier to kind of get your foot on the ground. And then once you're there, then you expand your indication profile to make money or hopefully help humanity, all that kind of stuff. So this is a graph that kind of highlights that what's the bottleneck in drug discovery. Right? So what's the success rate in the different phases, both preclinical and then in your clinical phases. And as you can see, the preclinical to phase 1 is 64% success rate. It's pretty easy if you have a good compound to get it to a phase 1 clinical trial. Now from phase 1, you may end up running into unanticipated toxicity and so on. About 44% go to phase 2. And this is then where the real drop-off happens. Only 20% of your phase 2 trials ever make it to phase 3. And phase 3 is actually pretty good. 65% actually then go to approval, and then 83% actually get approved for market. There's actually a number out there, I know it in my head, I don't know the source, but it says about a third of the molecules that ultimately make it to the market make their money back. So 2/3 do not. So again, this is not for the faint of heart, but we try because we want to improve lives, right? And then this is, and I'll share these slides on the website so you can, actually read them and pull them up on your computer later. This is a nice graph also from Nature Drug Discovery that highlights the cost of a drug per phase. And ultimately, when you take into account everything that doesn't work, right, it costs $1.8 billion to get a drug to market. Now, let's say you are the luckiest person in the planet, and whatever you pick from the get-go, everything works at the first try. It still costs, on average, $265 million. So this is, again, kind of circling back to the beginning of the presentation. This is why patents are so important, because no university or Southern Research or nonprofit organizations are going to be able to come up with $265 million. So you need to partner with somebody who has that amount of money. They're not going to invest into something unless they can make their money back, just because they need to stay in business. So you need to have your patent, because a patent allows you to prevent other people from doing the same thing, right? So then just briefly about where does the money come from? Who funds what part of this whole process? So on the left-hand side, the basic research, this is largely public funding like NIH, Department of Defense, has a lot of funds, things like that. Here on the right-hand side, when you're looking at clinical development and FDA approval, this is really where industry comes into play. But it's this kind of translational part that people call the valley of death. It's typically underfunded because it's not hypothesis-driven research. It's really difficult. It takes a lot of money. It takes specialized expertise, and it's kind of hard to do. So, yeah. So let me talk a little bit about how we can find money to kind of cross that valley of death. So there's a couple of opportunities at the National Institutes of Health that can help cross that valley. Of course, the majority of the NIH funding is still going to go to basic research, but there are specialized programs now that can help focus on that valley of death area. So here's a link you can click on whenever you get it. So NCATS is the newest institute or center at NIH, a National Center for Advancement of Translational Science. And NCATS funds the CCTS that we currently have here, Center for Clinical Translational Science. So they're really focusing on that translational piece from bench to bedside, and not just as a nice slogan, but actually doing it. So they have a couple of programs and open RFAs at different points in time, but one of two of their programs are called Bridging Interventional Development Gaps, or BRIDGES, or Therapeutics for Rare and Neglected Disease Trend Programs. Especially the latter, you can see how a big pharma company that's profit-driven, it's a for-profit company, won't necessarily go after an indication that has, I don't know, 600 patients worldwide, right? It doesn't make business sense. So this is where there's really a clear opportunity for government to step in, and let's, on a government funding basis, let's try to get these rare neglected diseases, get something for them to help them that is not related to trying to make money. The NCI, National Cancer Institute, they have a program called the Chemical Biology Consortium. You can have screening done through that program. We have actually one of our projects is doing a phase 1 clinical trial now at the clinical center at the NCI, and they pay for everything. They got the IND done all on their nickel. So you apply for it like a grant. You don't get the money directly to your lab, but they pay for outsourcing all those different pieces that you need to have happen. And then other institutes sometimes have particular drug discovery programs or RFAs, so you just kind of need to be on the lookout for them. But then a really nice resource too is foundations, and sometimes we forget about foundations as money pots. It sounds so negative, but opportunities to help get support, because they're typically really focused on getting something for a new disease, something new for their disease. For example, the Cystic Fibrosis Foundation, they're funding us now. We have a contract for several $1 million where we screen every compound in Southern Research's library for cystic fibrosis program. Michael J. Fox does a lot of things in the Parkinson's disease area. There's Alzheimer's Drug Discovery Foundation, Children's Tumor Foundation Drug Discovery Initiative. So whatever your favorite disease is, look for foundations, and there may very well be opportunities to actually do drug discovery. So then really briefly, in the last few minutes, I wanted to talk about what we have locally here on campus. So we have a collaboration between UAB and Southern Research. It's called the Alabama Drug Discovery Alliance. And it's really a partnership with the School of Medicine, the Cancer Center, and the CCTS together with Southern Research to kind of cover that whole spectrum of that pipeline. So the goal is we have so much research going on here at UAB, some at Southern Research as well, that let's harness that and let's develop new therapeutics, generate some new intellectual property, license, partner with companies, and get this moving. So the idea is, when you look at the left side, basic research, and the ultimate right side, clinical trials, that's really where UAB shines. And Southern Research, they have 7 FDA-approved drugs on the market. I mean, they know how to do this stuff, right? So the expertise in drug discovery and development, together, we really know how to do this if we partner and work together. So this is really kind of where our sweet spot is, again, in the valley of death, where we try to fund our programs. And what we do is we have an RFA once per year. We have a pre-application for 2 pages where we just kind of canvass the campus, like, hey, what you got? And then we select a subset of those, and we invite them for full proposals. And we look for, what's the scientific rationale? What's your target? It's going to be a really important driver. Have you defined your target, the molecular structure your drug is supposed to act on? And then what are some IP aspects, you know, things like that. So we have the science reviewed, we have our tech transfer office look at it, and then we have an advisory board that ultimately looks at the portfolio and they decide, okay, we want to go for this. So then what's kind of used to be unique, now it's being utilized a little bit more on campus, we put project teams together around each of these projects. This is not just a PI in his or her own lab doing things by themselves. This is a team sport, so we get everybody together regularly, every 3 or 4 months, talk about where are we, what do we need to do, have we run into any trouble, hey, let's brainstorm, troubleshoot, and it's usually a lot of fun to have those meetings. So we have people from Southern Research, chemists, high throughput screening people, pharmacologists, etc., clinicians from UAB, and we all get together and we kind of make a plan to make things happen. This is our advisory board. You can look at what fancy people we have on there, all smart. So like I said, we have these project teams built around them, and just to kind of help the project to stay on track and not go off like, oh, this is an interesting result. Let's, you know, go down a rabbit hole. Well, no, get back here. Let's focus on the next milestone. So ADDA really covers high throughput screening, assay development, and the actual screen, and then some medicinal chemistry as well. And then once we get to, you know, development where you need to start talking about formulation, toxicology studies, and so on, that's usually where we would probably want to partner with somebody who has deeper pockets and things like that. So we give $50,000 per year for 2 years to the lab, and that covers whatever happens in the lab. And Southern Research pays for everything that happens at Southern Research, so the screen, chemistry, and then we also now have resources. We have a philanthropy donation of several $1 million that was made to us to help move these programs forward. So we try to typically use that money for chemistry programs, or we really need to put a couple of chemists on the program and make, you know, a couple of analogs a week, so maybe 20 every 2 or 3 weeks, and test them and go back and test them, etc., etc. So we jointly own any IP that comes out of these programs. So it's a really nice program. So this is our current pipeline. We have 3 CNS programs. We have a lot of oncology programs just because we have so much oncology research going on. We have 2 antibacterial projects, and then we have a couple of one-offs. And you can see a lot of them are early because we have an RFA every year, so we have a couple starting at the beginning every year. But then we also have one that's now phase 1 clinical trial, so it's really exciting.
So how many of those first-year projects get funded this cycle?
2 or 3 per year. And when we first started, we had 2 RFAs per year, and so because we kind of wanted to prime the pump, so we had about 6 start for the first 6 per year for the first 2 to 3 years. What we also found, though, is the first couple of years there was not a lot of sophistication yet on everybody's part, including mine. So it took a while from the start of the project until something was ready for high throughput screening. Then we got smarter and smarter, and we talked to a lot of people a lot more. So the people that came in maybe 2 or 3 years later, they were ready for high throughput screening in 3 months. So all of a sudden we had a bit of a bottleneck because everybody was ready for high throughput screening at the same time. So it was 1 year, and I think Bob will have a slide on it when he talks about high throughput screening, when he screens several million compounds for ADDA projects, because he was trying to catch up. And now we're finally caught up. About last year where we're kind of back into the 2 or 3 per year. So there's not a backlog anymore. Fine.
So the oncology drug that's in phase 1, how long did it take to get to phase 1?
Well, that one kind of cheated. so that the phase 1 program is a compound that actually came out of the Southern Research's freezers. So it was already, what it came in on is that we funded a critical animal experiment to show that it worked through the mechanism that we thought it did. So it was one of those nucleotides that was developed as an anti-cancer drug, just for, you know, it was very crude, you know, 20, 30 years ago. You just want to kill cancer cells, and hopefully your healthy cells don't kill as fast, right? So this compound was developed for that particular mechanism, but it actually didn't work that well that way. What we found out, you know, years and years later, was it actually was kind of changing how the methylation patterns in cancer cells. And it's kind of that field didn't really exist even when this compound was first discovered. So when it was pulled from the freezers, and I don't know how they thought to test this. They figured out that it inhibited this DNA methylation process. So what we then did is came in as an animal experiment. Let's see if it actually treats this tumor in animals, which it did. And then we went to the NCI and they paid for everything else. So because it came in at the later stage, it was a pretty quick jump to phase 1 clinical trials. Let's see, the one that started from scratch and it's the furthest is probably our LRRK2 program. It's a Parkinson's disease program that started from absolutely nothing to now being in lead development. This was started in 2010, so 7 years. So you can see this takes a long time. Now hopefully, we like to think we've gotten better at this, so maybe the ones that start, just started last year, they may do it in 5 years. I mean, who knows? But it is a lot of fun usually because everybody learns something in these teams. You have the absolute experts on a certain molecule in like Parkinson's or diabetes or whatnot, working together with the chemists who don't know anything about the biology. Biologist doesn't know anything about the chemistry. And then my job is to make them understand each other. And then you come up with a plan to get it forward and you have these team dynamics and everybody just is really excited because everybody learned something new. And you really see the progress going on and everybody gets really excited about it. So, all right, so that's it for today. Any more questions? Let me see if there's any questions from the virtual audience. No bubbles have come to, oh, look, there's a question. Are ADME studies necessary when a drug is metabolized by a gut enzyme and not in the liver? Yes. So ADME, sometimes we just think of the liver as the clearinghouse for whatever happens in the body, but actually your whole body metabolizes this drug, and some drugs are even, you breathe them out. Some get excreted in mother's milk, some in saliva or in the sweat glands. So you really need to get the whole picture really as broad as you can. So you start usually with liver because that's where the bulk of things happen. And if something in vitro, so just in a cell culture system with microsomes or with hepatocytes, gets chewed up by liver cells really quickly, then you probably wouldn't even go into a whole animal. Now the reverse, when it doesn't get metabolized by the liver in vitro, so many times when you give it to an animal, you see it does disappear within minutes after all, and you're like, what? How does this happen? Well, it's probably because something else besides the liver did all the metabolizing for us, or maybe it gets excreted in the urine, things like that. So yeah, you need to do ADME studies for anything, no matter where it gets metabolized. All right, thanks for your question. All right, I will put these slides up on the ADDA website. If you just Google ADDA and UAB, it'll pop up. I don't know the website either, so off the top of my head. And you probably wouldn't remember if I told you right now anyway. Let's see, I think my name, nope, sorry. If you want any more questions, if you have any more questions, you can always send me an email at addda@uab.edu. Or Micah, my first name at uab.edu, but you probably will forget what order the vowels need to go in. So ADDA is easier to understand. It goes to me as well as the same inbox. And I hope to see many of you next week. And thanks everybody online. And hopefully it was understandable even from a remote location. All right, thanks guys.